Welcome to our first clinical genomics webinar, and thank you all for being here. We are very excited to start this series about clinical genetic research from leading scientists in the field. My name is Mariam. I am the Science Communication and Marketing Officer, and today I will be hosting our first speaker, Dr. Mohammed Uddin. Dr. Uddin is the founder of GenomeArc. His research is focused on human genetics and neurodevelopmental disorders. He identified numerous genes and variants associated with autism and epilepsy, and his talk today will focus on genomic interpretation and how it can transform patients' lives. It's a very interesting topic that will give us insights on how genetics can affect patients' outcomes. And now, without further ado, let's get started. Thank you for joining us today, Dr. Uddin.
Thank you, Mariam, for your kind introduction. I'm going to talk about how genome interpretation can transform patient lives. I'm going to speak a lot of overview or summary results due to the time restriction, so apology in advance for the brevity. So rare disorders are a collection of conditions that impact developmental milestones and manifest very early. 1 in every 8 individuals affected by a form of rare conditions. These conditions manifest due to mutation in a single gene. There is a small subset of mutations that are recurrent, that appears in unrelated individuals. Recurrent means it happens in the same location of the genome. Most of these pathogenic mutations are sporadic and individual specific. It paints a complex picture for diagnosis trajectory and for therapeutics. There are over 6,000 of rare conditions and traits reported to date. Over 4,000 unique genes been reported to have pathogenic mutations. All types of putative mutations have been reported as causal for numerous single gene disorders. The most daunting mutations are these missense mutations, where a substitution often present a complex scenario to establish disease association. There are biallelic mutation, a compound heterozygous mutation also been reported, and for population that have consanguinity or founder effect also suffer from a loss of heterozygosity that produce a lot of homozygous state of variants. The origin of single gene disorders mutation have both sides of inheritance complexities. Some mutation appear to be as de novo, and these mutations originate at the germinal cells of the parent. So the zygote apparently inherits this mutation from the spermatocyte or oocyte. So the origin of these mutations are at the oocyte or spermatocyte DNA repairs or DNA mismatch that produces the mutation. In other scenarios, mutations are inherited from one or both parents. If it is a recessive condition due to homozygous mutation, both parents will be carrier. A lot of rare disorders are reported to be recessive. Or one parent might also carry a pathogenic mutation, but due to incomplete penetrance, the parents might not show any phenotype, and the kid, the offspring, might manifest a severe condition. And if you look at the variant types from all length spectrum, from one base pair mutation like missense or spliced mutation to karyotype, where we can see mutation at the scale of chromosome can be impacting a condition, rare genetic condition. So it's not converging into one type of variants, rather all type of variants we see that been reported to these rare conditions. Most of these variants are located within the coding region of the genome, making the protein disrupted in structural folding that loss or gain function. The protein can lose its known function and gain something absolutely new. Due to this convenience of the coding region, exome sequencing is the most adopted genome sequencing approach in the clinic that can identify most of the pathogenic mutation that we see in single gene disorders. Clinical exome is the commonly used and more equitable globally. Slowly, clinics and hospitals are adopting whole genome sequencing to look beyond the coding region and move into those uncharted territories of non-coding variant association. We still have not figured out how to how to derive association from non-coding variants and elements of, and then relate it back to the rare conditions that we see. Whole genome sequencing also helps profiling the full spectrum of mutations, especially provides base level resolution for structural variation detection to make clinical genetic test equitable and globally adaptable. Exome test still is the most cost efficient and provides the most amount of clinical yield or diagnostic yield. So now I want to shift gear towards multigenic or multifactorial rare disorders. And if you look at that category of rare disorders, neurodevelopmental disorders is one of the most prominent type of rare disorders that affects a lot of kids. So kids with autism spectrum disorders or intellectual disabilities or early onset epilepsy encephalopathy are one of the most common types of neurological or neuropsychiatric diseases that impacts more than 3% of a given population. From mono and dizygotic twin studies, we know that these conditions are extremely genetic. So what it means is there's a little room for environment to cause these diseases. So mostly genetic variants should be able to explain most of these cases. Moreover, these cases are extremely heterogeneous in phenotypes. They also have overlapping phenotypic manifestations. So if you look at the neurodevelopmental disorders in terms of the functional aspects and the structural aspects, you can see there are a lot of these conditions that can be categorized as just only functional, and you see these conditions only manifest due to the dysfunction at the level of synapses. And these dysfunctional diseases involves autism, epilepsy, encephalopathy, ADHD, schizophrenia, some form of intellectual disabilities, and obsessive compulsive disorders. And the other spectrum where we see functional deficits along with structural changes, that includes micro and macrocephaly, focal cortical dysplasia, double cortex syndrome, and some form of epilepsy encephalopathy that also shows some dysplastic tissue that brings structural changes in the brain. And so far, our lab and others across the globe mapped over 100 genes that are putative and causal for autism spectrum disorders. So similar kind of map exists for epilepsy and intellectual disabilities. Although these projects involve many years of technological advancements and numerous novel gene detection for autism and other diseases, these genes do not converge into a common pathway. So it means these genes that we find that are causal in kids with autism not necessarily manifest the disease in the same pathway, that, or the etiology is not the same. So the etiology of ASD is still not clear due to lack of genomic and molecular convergence. So the projects across the globe, they're ramping up recruitment and doing more sequencing and applying more analysis to characterize the observed phenotype and the observed heterogeneity at the level of DNA. In a recent study from our lab, we have shown significant overlap of genomic loci between NDD, neurodevelopmental disorders, and congenital anomalies. Congenital anomalies refers to any congenital abnormalities that we have observed in those kids. For our cohort, we identified 18% of de novo structural variants in NDD and 40% of de novo structural variants within congenital anomalies overlaps in genes that are developmentally constrained. So we have applied the platform Horizon from GenomeArc to assess pathogenicity of structural variants. Horizon provides multiple lines of evidences to support pathogenicity for structural variants. Using Horizon annotation, we have identified great degree of genomic overlap between NDD and CA cases, and these de novo variants overlap of structural variants observed in both male and female with great significance. And this cohort, actually, we're still analyzing it to pinpoint why these genes are overlap between these two conditions and provide totally two different types of diseases. It's a big puzzle to solve. And I want to give you an example where, having this heterogeneity in mind, we mapped these variants, deletion and duplication, into a gene called EHMT1. And this gene been reported with variants impacting numerous exons of the gene. If you can look at the figure, the blue are the duplication, the red are the deletions impacting numerous sites of the gene. And it produces roughly around 77% NDD cases. So most of the patients that we see have NDDs, but then there are small portion of 9% cases that only have congenital anomalies. And there are also 14% of cases only have neurodevelopmental disorders. And within those 77%, there are also report of having both NDD and congenital anomalies. So it brings a huge complexity why NDD and congenital anomalies manifest together, or sometimes it doesn't manifest together, even though the causal variance is the same, impacting the same gene. So our study is not the only study that kind of identified this kind of complexities. We are just one of the many. A number of studies around the world observe this heterogeneity across different types of neurodevelopmental and rare disorder genes. So confirming variant pathogenicity according to ACMG guideline, variant can be classified into three main categories. So pathogenic, benign, and variant of uncertain significance. While we observe this heterogeneity within the pathogenic variants in our last study, or in the last slide, the most daunting task in clinical genetics now is to interpret variant of uncertain significance. These variants are extremely rare, but lacks enough evidence to make them pathogenic. So unfortunately, we have thousands of these variants of uncertain significance in numerous rare disorders, including neurodevelopmental disorders. At least 30% of these neurodevelopmental disorders carry a VUS. So I want to show you an example where in our lab, we are trying to resolve such a variant of uncertain significance impacting a gene called PPP1RNA that encodes neurabin protein. So these are numerous, these numerous variants that impacting these genes have been kind of classified as VUS. So our functional analysis show neurabin binds to actin filament and have cross-linking abilities. Neurabin involves neurotransmitter regulation, play important roles in dendritic spine morphogenesis and neurite formation. Neurabin also shown to be regulating synaptic assemblies. We have initially conducted qPCR analysis testing the common mRNA sequence across different human tissue types. And it shows upregulation bias with the brain tissue. So our primary objective for this study is to characterize and establish the role of neurabin into the pathogenesis of NDD that we observe at least for these kids. So what we have created is a CRISPR-edited cellular model to induce one base pair insertion within the cassette of neurabin, and one base pair insertion actually truncates the whole protein. And if you look at the Western blot image, that it shows a heterozygous kind of band of the protein. And after producing the homogeneous line for the mutant CRISPR-edited cell lines, we have confirmed the heterogeneous mutant line, and it is kind of reflective of the mutation that we see in the patients, because the patients are impacted with heterozygous mutation rather than homozygous mutation, which is extremely hard to model, because homozygous mutation, you have both non-functional protein, but heterozygous, you have one functional and one non-functional protein of the same gene. So when we have the homozygous line, so the homozygous line refers to all cells have that heterozygous mutations, no other mutation from the CRISPR. So the human iPSC derived cell lines have been then converted into neural progenitor and later into mature neurons. The developed protocol produces functional mature neurons within 4 weeks. Numerous neural progenitor and neuron specific markers have been stained to confirm the differentiation that we have conducted, and you can see there are visible dendrites marks within neuron soma, and these dendrites are connected through synapses with other neurons, and these mature neurons represent a very viable model of the mutant lines for further phenotypic analysis downstream. So we have done iso-seq. That is a very new technique to find molecular phenotype. This iso-seq kind of sequence of full-length mRNA. Instead of using short read, we have applied long read technology because this gene have multiple transcripts that we have observed. And what we have observed is that due to the CRISPR-induced mutation, the mutant neurons lost more than half of the mRNA that didn't get regulated, and we also see a loss in diversity of the isoform. Some of the isoform that are present in the wild type is not present in the mutant neurons. We also generated a line where we took the mutant line and overexpressed with the wild type neurabin just to see if it can reverse the course of molecular regulation. And you can see the middle graph of the graph B, it shows the isoform that we lost in the mutant. Actually, it came back, but not with all type of isoform that we saw in the wild type. And interestingly, the mutant cell line kind of acquire a new transcript that is not present in the wild type, or the mutant cell line that is overexpressed with wild type neurabin also doesn't have that acquired transcript that we have seen in the mutant lines. So this kind of shows that this one base pair mutation actually dysregulates the splicing mechanism and the regulatory machineries for neurabin. So we are conducting many new analysis to identify the morphological differences between the mutant and wild type neurons, and also the synaptic densities and synaptic functions between the wild type and mutant cell lines that we have generated. And it will give us functional insight how this mutation can impact the neurons of the patient. So now, just to be on time with my talk, so I just want to touch base some of the future techniques or technologies that will create great impact in clinical genomics. So the mass scale modeling of this VUS will be happening soon because of this CRISPR or gene therapy like technologies. And machine learning is another technique that will perform better in predicting variant effects if we have enough training data. We're already using machine learning variant detection tools like deep variant that outperforms all other algorithms. Rapid genome sequencing. So now I want to talk about the emerging technologies that are about to impact clinical genomics. There are multiple emerging technologies that will create great positive impact in clinical genomics. Mass scale modeling through CRISPR or small molecule gene therapy kind of technology will affect heavily to characterize or resolve this variant of uncertain significance. So it will increase the clinical yield in near future. Another technology is machine learning or artificial intelligence. So it will be able to predict the variant effect if we provide or train the model with the right kind of tag data. So we are already using machine learning variant detection tools like deep variant that outperforms all the other algorithms out there. Rapid whole genome sequencing is another amazing approach to save NICU and PICU patients, the critical care kids. A good portion of these patients are now shown to have a genetic cause, and rapid genome sequencing is necessary due to diagnostic time constraint. And a platform like Horizon is essential to predict variant pathogenicity with ultra-fast analysis. So clinical interpretation can be done in a faster turnaround time, and clinician can make a timely diagnosis and intervene for therapeutic options. So single cell sequencing or single cell omics actually is a very close to my heart kind of technology, although I'm not going to talk about it. It's changing everything in medicine, the way we look at it. But I want to talk about our emphasis on the topic of pangenome that involves long-read sequencing. So long-read sequencing recently closed the 8% gap that was left out by the reference genome. And the first telomere to telomere genome was published last year. I've been fortunate to be part of the HPRC now. We are providing huge long-read genome sequencing data to construct numerous population genome. And now we have seen a huge influx of paper that shows de novo approach is becoming more applicable to detect variants that were never been seen or detected using reference genome. So long-read sequencing can reach the complex region of the genome that has never been touched by the short reads. It will fill out those sequencing gaps that left out by the short read technologies. These gaps are bringing new types of variants like tandem repeats or mobile elements that can be assessed for pathogenicity with rare disorder or any disorder that we want to assess for. This tandem repeats and mobile elements never been identified with great confidence with short read technologies. So now we can detect it great confidence and measure its association with numerous diseases. And long read also provides a markedly different kind of benefit compared to short read. It can phase the genome and it can identify the origin of the variants in terms of parents. And long read also can identify the gene duplication complexity that we have in the genome, which never been been touched with short read sequencing. So pangenome, when we do long read sequencing, pangenome construction become a viable kind of option for us. Pangenome is a reference set of haplotypes that were constructed from high quality genome assemblies from long read sequencing. Recently, Human Pangenome Reference Consortium constructed pangenome involving numerous ethnicities, and these ethnicities represents their own kind of pangenome. The pangenome usually can be represented with graphs. The graph genome takes individual haplotypes. If you look at the bar graph here, it shows each individual have two different haplotypes, and each of these haplotypes can be used to construct a pangenome. So the next diagram down below, it shows two different haplotypes, one in blue, one in yellow. And whenever there is a variation between the haplotypes, it can create some different edge and nodes. So you kind of think of like inserting 3 billion base pair haplotypes, two of these haplotypes from each of the individual, and creating this huge graph genome. And you can traverse the graph genome to identify the overall variants observed within a set of assemblies and without the help of a reference genome. And pangenome construction by the HPRC group involved 47 phase diploid assemblies that added 119 million base pair of a chromatic polymorphic sequence and about 1,100 gene duplications relative to the existing HG38 reference genome. And roughly 90 million of this additional base pair that has been added are coming from the structural variants. And 2 weeks ago, the Chinese came out with their own pangenome that involves 36 ethnic minorities within China, and they did 58 phase diploid assemblies, identifying 5.9 million small variants that are not part of HPRC pangenome, and 34,000 structural variants that's also not been reported in HPRC. So these 5 million, so almost 6 million small variants and 34,000 structural variants never been seen before. You will not see it if you use HG38 reference genome. So it's a huge amount of variants that will be missed if you use HD38. So this pangenome is becoming the next big thing for clinical genetics as well, to identify those variants that have never been reached out using traditional short read based and genome reference based approaches. And the other interesting thing is if pangenome also can identify the sequence that are missing within a population. So the missing sequence in Chinese population includes archaic derived alleles and genes that confer essential functions related to keratinization, response to ultraviolet radiation. Also, these genes involves in DNA repair, immunological responses, and lifespan, so longevity. So this is kind of the impact that pangenome can bring into. So this is a huge plethora of variants that never been, assessed genotyped in different patient groups for different numerous diseases. So that's what we'll be doing next. So I would like to thank all the lab members and all the collaborators for making this happen, and all these experiments that we have done throughout the years only been possible because I have a great team of postdocs, PhD students, and master students who are dedicating their time to conducting these amazing experiments. So thank you, GenomeArc, for inviting, and thank you for connecting with clinical genomics. I hope that we have learned something about the emerging technologies and how it might impact the future of clinical genetics. Thanks.
Thank you, Dr. Uddin, for this amazing talk. And now for some questions. First question, what is the impact of long-read sequencing in clinical genetics?
Thank you for the question. So long-read sequencing will have a long-lasting effect. I think the whole clinical genetics is moving from short-read sequencing to long-read sequencing because of the added benefits. So it can phase out the genome, without too much effort. Because if you look at the short read, how you want to phase genome, it's a very complex problem. But this technological advancement of long read made it possible. So if you genome, if you sequence the genome of a kid, you essentially can identify genome information of his or her parents. So that kind of give you added benefit of not doing trio analysis for everybody, I guess. So in clinical setting, it will contribute a lot. It will identify, all these inherited variants from different parents, and it will bring a lot of diagnostic yield that was not been possible before. And as I said in my talk, so there is a lot of variants that tandem repeat or mobile element variants that needs to be probed or assessed in terms of their disease association. So I'm thinking of that these tandem repeats or mobile elements will resolve a good subset of these neurodevelopmental disorder cases, which are already known, like Fragile X syndrome, known to have tandem repeats. There are more of these other diseases, lots of epilepsy. Few cases in autism also have this tandem repeats and mobile elements association, but it's not been captured well due to the technological limitations. Okay, so that is coming, and also long read technology is the core for pangenome, so that will still remain very relevant.
For the second question, well, how do you think artificial intelligence should be adopted within the framework of genomics, especially in clinics and hospitals?
Thanks for the question. So yeah, artificial intelligence, it's going to be a big, technology for a lot of different types of problem. And in medicine, especially in genomics, there are problems that it can be adopted. But I'm not going to hype up that it will solve everything, because machine learning really excel in image-related problems. So if you see how machine learning is impacting genomics, the most relevant tool from machine learning perspective is for example, deep variant, and nanopore technology uses those electronic signals to identify the bases, or to call the bases from the electronic signals. So both of these being converted, this problem has been converted from quantitative data to actually image problem, because machine learning is amazing in image deciphering and identifying patterns from images. So far, any problem can be converted into image kind of pattern detection. Machine learning can do a good job, but we have to see in other types of problem, like optimization problem, that is riddled, in genomic medicine. We have to see how that happens. But machine learning so far works better if you have a good set of training data, because this is not an unsupervised approach. This is a supervised approach that can give you a good amount of optimum solution if you have the right training data that you have trained the model with. So I would say just data is the key here for machine learning to impact genomics. But rare disorder is really hard, because you can train, but not many rare disorder have a repetitive kind of phenotype or the genome, right? So it's very rare to have multiple rare individuals with similar kind of genomic background or phenotypic background. So training with rare disorder is extremely hard. It's a challenge that we have to somehow overcome through collaboration. And also look beyond machine learning. So because I think the language model is not that effective in deciphering kind of the phenotype well enough. So we saw like Horizon platform does a great job kind of linking the phenotype with the genotype, but machine learning itself is not enough. You need all sorts of other regression model or other computational model algorithms, maybe deterministic, that are known to perform well along with machine learning. So we need a kind of like a blend of different types of algorithm to identify what works better. It's not going to be all machine learning all the way, but it is a strong technology. We have seen it outperforming many existing tools that uses deterministic kind of algorithm.
Third question. There are multiple pangenome initiatives that provide reference genomes for their own populations. Do you think this is the future of clinical genomics, to identify pathogenicity of all variants?
Absolutely, yes. So the pangenome question is very relevant to the clinical genetics we want to do in future, because pangenome, again, I have to bring back the long-read sequencing. It kind of identifies all these interesting regions that was left out from the reference genome, and it brings a plethora of variants. If you look at the Chinese initiative, they just did 50 plus genome assemblies to construct their pangenome, and they identified about 6 million small variants that was not part of the HG38 genome, and also 30 plus thousands of structural variants that was not part of his HPRC or the HG38 genome. So if you want to get a full spectrum or want to identify all the genetic causes, you have to identify all the variants. So it seems like the current reference genome have huge limitation in terms of ethnic background wise, or even geographically, population might be different. For example, Newfoundland is a founder population. They started the population with few families from Scotland or Ireland that moved into this island, and huge inbreeding happened, and the current population have sequences or variants that are rare in other population but common in that population. So there might be sequences that there will not be found in the reference genome or in other pangenome. So they need their own reference or pangenome. So similarly with different ethnic minorities across the globe, they need to bring this long-read technologies into the clinic to create a healthy reference genome, so it can be used for clinical causes, because those variants that they will detect from pangenome construction will not be detected. A lot of those variants will not be detected from this traditional HT38. And we can see we reach almost a plateau in a rare disorder diagnostic yield. So this pangenome can break that plateau and bring interesting, diagnostic yields that also will lead to different pathways for therapeutics and save lives. So we need to interpret genome in terms of pangenome and not in terms of reference. That is the next big thing in clinical genetics.
Thank you, Dr. Uddin, for joining us, and thank you all for being here. Stay tuned for more exciting talks on clinical genetics. We look forward to you joining us.